Tak, dobré odpoledne, vážení přátelé. Dnes padla měla povinnost vádět dnešní přednášku. Napřed teda dle tradičního zvyku čtvrtečních setkání anonce příští přednášky. To bude přednáška Petra Mikuly Neboj se, čili jak prax člověka ovlivňuje chování zvířat. Tak, now we can switch in English. And I would, as the old eyewitness person, remember that we have now already 70th semester of our biological Thursday meetings in this traditional room where Einstein's and so lecture. During these 70 semesters, we could host about almost 300 very biologists, including the leading authorities of contemporary biology, like Bert Miller, Günther Wagner, Stuart Kaufman, James Brown, Robert Ricklefs, and so on and so on. And today,
don't make me more nervous.
Opportunity to enlarge our list of the prominent top biologists with N. Huysseune. And Huysseune is an emeritus professor and past director of the head of the Biological Institute of the University of Kent, which is of course a very important influencer institution with more than 200 persons in staff, and was more than 12 years head of that institute until her retirement. And she is, among other, one, I would stand founding father, but it was not necessarily a founding father, of evo-devo, evolutionary developmental biology, the branch of the contemporary biology which integrate the issues of genome and molecular biology to organismic perspectives, and for instance organized the second Congress of Evolutionary Developmental Biology in Ghent. She contributed the topics from beginning of her scientific career with important discoveries concerning dynamics of the mineralized tissues of vertebrates, what happened between cartilage and bones and so on, and especially teeth and dentitions, which is of course essential apomorphy of vertebrates which mediated conserve ecological prosperity of our clade. She published about 200 of important papers on that topics in most prominent journals. She was appointed to be keynote speaker of many congresses and so on. She acted as the president of the International Society of Vertebrate Morphology and organized as the president a very successful congress in Prague, and so on and so on. Was mentioning that several years ago and spent some time here in laboratory of Robert Cherny, her sabbatical, and now after retirement he took even a full position in that laboratory. They are very proud that so important scientist is just a member of our department. We are looking forward to her lecture, and please start.
Thank you very much, Ivan. That was a very nice introduction. I was told this is a very prestigious series, as you also mentioned, so I got more and more nervous, and I hope I will not disappoint you. Yeah, so also to the organizers, of course, thank you very much for giving me this opportunity to talk here. Okay, I have this somewhat weird title, A Mouthful of Teeth. And what do I mean by this? What do I mean by this? What is not going here? Somebody?
To the right.
Maybe we are a bit far away. Okay. Yeah. All right. So the title, admittedly, is taken from something an expression in my native language, Dutch, and what it means is that it indicates that you run out of things to say, you run out of ideas. And if you look at this mouth, this beautiful mouth with teeth, the question is, of course, do you know where these teeth came from? Do you know where your teeth came from? Probably a number of you have now a mouthful of teeth, right? So that is one aspect I will be discussing in this presentation, is the evolutionary origin of the dentition. And the second meaning of a mouthful is it means as much as a mouth will hold. And you have this picture here of an anglerfish full of teeth in the mouth, really a mouthful of teeth. I used this picture just to show you, and what I will show you also later in the presentation, is that let's say in ancestral gnatostomes, or even at the basis of many lineages, we have species that have teeth really all over the place, all over the mouth and the pharynx, and I will comment more on this later on. So first, something about the evolutionary origin of teeth. And before so I was told this is a very broad audience, and I thought it was relevant to introduce a little bit some terms or concepts. So if we have here a lamprey, and if we make a horizontal section in this direction, and we make a cartoon out of that, we see a number of structures which will be important for my presentation. So you have here, of course, the mouth entrance, which then continues in the alimentary canal, and the skin is on the outside. And this is a good cartoon to introduce you to germ layers, which will be important in my talk. That is the ectoderm on the outside, shown in blue here, and the endoderm here shown in green. Now, I know this is quite upsetting for some people to have the endoderm in green. The reason why it's not yellow, as it should be, is just because the background is white and you don't see the yellow very well then. So please accept that for the time being, my endoderm is in green, okay? Another thing I want to point out on this cartoon is these gill slits that you can also see here. So they are open communication channels between the pharynx and the outside environment. Now, if we move then to a gnatostome, like here this carp, and we make a similar section, it looks a little bit different. Again, we see the mouth here, the pharynx, the lining of the pharynx, the mouth. The lining of the mouth is partly in ectoderm still. The rest of the pharynx is lined in endoderm. And one important difference is, of course, the operculum, which is a development from the second arch. These are the successive pharyngeal arches, of course, the gill slits. And the little spikes you see are representative for the teeth that can sit in the mouth, but also in the entire pharynx. So with that, I have introduced you a number of terms. And now moving on to the origin of teeth. Now for quite some time, I would say certainly 100 years, The canonical view, so the generally accepted view about the origin of teeth, was that they derived from skin denticles or dermal denticles. And there are a number of arguments for this. For example, in agnatins, so the jawless fishes, the jawless vertebrates, many of them had this extensive dermal armor, for example, here in this anaspis. And you see a number of scales with tubercles, and if you look at these tubercles, the structure, the composition of these tubercles is very reminiscent of that of teeth. You see bone, yes, but you see also on top of them tissues that we also find in teeth. A hypermineralized layer of an enamel-like tissue, enameloid, or these are tubercles of related species where you can have a dentine layer between the hypermineralized and the bone layer, or even an extensive dentine layer. So these structural similarities between these tubercles or denticles in agnatins have supported this canonical view on the origin. So apart from structural similarities, there's also developmental arguments. For example, here below you see sections through the lower jaw of a recent shark, and these are the developing teeth here. These are developing dermal denticles, or placoid scales as they are also called. And of course, again, you see a high similarity between these two structures. So the idea is that dermal denticles at the dawn of gnatostomes moved from the outside, from the skin, into the mouth, represented by this arrow, and this theory became known as outside in. However, so that was for a long time an accepted view, but then in the late, in the late 1990s, the view was challenged by a number of authors, and they claimed that, in fact, teeth are not derived from outside. They are derived from deep inside the pharynx, so they are derived from endoderm, and in addition, they would have originated prior to the origin of jaws. Now, the authors had a number of arguments, paleontological arguments and neontological arguments. I'm just presenting two. One of them was the existence of conodonts. Conodonts were seen as the first vertebrates with mineralized structures, and these elements were likened to teeth. And another argument was the finding of telodons, like this guy here, with pharyngeal denticles. Pharyngeal denticles, as shown here and as represented in these two drawings. And according to the authors, the arrangement of these pharyngeal denticles in this kind of spiral shape, in this whirl-like shape, pretty much resembled chondrichthian or ancestral Paleozoic and chondrichthian tooth whorls. So a number of paleontological arguments, also neontological arguments, to argue that teeth develop deep inside the pharynx and then somehow spread out in the mouth. This view became known as the inside out. Not that the teeth got out of the mouth, but as opposed to outside in. Okay? This view, again, in the late 1990s, it was heavily challenged, but it was also a good way to reinvestigate all the arguments that were used for each of the two views. So that was actually a good thing. And a number of years after the initial proposition, and now a number of years ago, in our team, we suggested that we should go back to the canonical view, but in a slightly modified way. We argued that the teeth need indeed ectoderm, but different from the regular outside in, from the canonical view, we suggested that this ectoderm may not only come via the mouth, but also via the gill slits, which of course would make it much more logical to explain the existence of pharyngeal denticles. And perhaps, yes, perhaps teeth originated prior to jaws, although this is contested. Anyway, what were our arguments? Also arguments from paleontology. We, scrolling through the literature, we observed that pharyngeal denticles or teeth are present only in fossils also known to possess gill slits. Remember that we attached a great deal of importance to this passageway for the ectoderm. That internal denticles or teeth are found only where there's an opening to the exterior, for example, the spiraculum. And for example, here you have a section through coelacanth, latimeria, at the level of the spiracular pouch. And you see here these patches of spiracular teeth. And then the third argument was that pharyngeal denticles and teeth disappear in early tetrapods with the closure of the gill slits. Now, there are quite some fossils, temnospondyle amphibians, that still have these branchial denticulated plates, and paleontologists, I hope I'm not wrong with the paleontologists here in the room, paleontologists tend to use the presence of these branchial ossicles or denticulated plates as evidence for open gill slits. So that fits nicely with this modified version of the outside in, right? Okay, now, this is a schematic from a paper in which Robert actually participated that very nicely I also changed the colors a bit, but it very nicely summarizes what I have told you so far. So the outside in assumes that dermal denticles moved via the mouth to produce the teeth. The inside out does the opposite. The teeth developed deep in the pharynx and moved forward. And then the modified outside in has access via both the mouth and the gill slits, but it would be the ectoderm that contributes to the teeth. Okay. Now, one of the arguments of the challenging view, the inside out, was that there was never ever an evidence that for a transitional stage between the dermal denticles and the teeth. I showed you the shark pictures, but the claim was that there is no transition between the two. And in quite recent years, in 2020, Donglei Chen and her colleagues published a paper on stem ostichtien, Lophosteus, where they showed, and they used very sophisticated imaging using synchrotron, they could actually remove this argument because they saw individual what they call odontodes, so these dental units. They observed that they form sequentially to the outside producing dermal denticles and to the inside producing teeth, which are next also replaced. Okay, now before I continue along these lines, it may be wise to just briefly introduce you in how a tooth forms, because you have to know this to understand the rest of the story. So very briefly, a tooth is the product of a common, let's say, common action of two soft tissues. You need two soft tissues to produce a tooth. You need an epithelium and you need a mesenchyme. And the mesenchyme is called ectomesenchyme because it comes from the neural crest. But other than that, the formation of a tooth is a very conserved thing. Whether you look at a little teleost fish like a zebrafish, or you look at a mouse. So you have an epithelium that starts to fold. So the epithelium is shown here in light purple. The darker purple is the part that participates in the tooth. And so you have a number of morphogenetic movements in these soft tissues. And then at the contact zone between the mesenchyme and the epithelium, finally the hard tissues of the tooth are formed. Okay, now returning to this picture that I just showed you, we are not, or we never claimed that it is the complete denticles, the complete odontodes that sort of move into the mouth, sort of march into the mouth. No, what we suggest is that it is competent epithelium from the ectoderm that would move into the mouth. Okay, so the question is, how could external epithelia have transferred the competence to form teeth into the mouth, into the pharynx? So how would they have transferred this competence to internal epithelia? That is a question that we have asked ourselves for some while, and we have engaged in this study. Not by using fossils, because obviously you are limited with the experiments there. So we had to turn to an extant species, and we used zebrafish to examine this question, to test the role of external epithelia in tooth initiation. And now zebrafish is actually, excuse me, zebrafish is actually advantageous because it has pharyngeal teeth. In fact, it has only pharyngeal teeth. It doesn't have oral teeth, that you may know. Zebrafish is the same family as a carp. I know Czech people are very familiar with carp. So you all know that you cannot be bitten by a carp, right? Because they have no oral teeth. Well, a zebrafish has no oral teeth either. So pharyngeal teeth. And this is an early stage of development, 5 days post-fertilization. Zebrafish develops very quickly. And you can see here on the last pharyngeal arch, these are all the pharyngeal arches, the mandibular, hyoid, and then 5 branchial arches. And the last pharyngeal arch carries these teeth. Mind you, this is very small. I'm talking here about teeth that are maybe 12 micrometer across, 50 micrometer high. Very small. But they are teeth. They are full-blown teeth. If you look at an adult, on the other hand, we still have the pharyngeal teeth here on the last arch. It's a bit more elaborate now, and it's a very stereotypical pattern, which is also very convenient if we do experiments. We know what to expect, yeah? We know exactly how many teeth, when, where, and so on. This is just a scanning picture of one of these sets of pharyngeal teeth on one side of the animal. Okay. Oops. Sorry, what's happening now?
Next one, the arrow to the right.
Okay. So I return to a cartoon that is very similar to what I showed you before. So we have here an embryonic zebrafish. We make a section in this horizontal direction. And a cartoon of this shows us here, it's very schematized, of course, the mouth, the pharynx lined by endoderm, the floor and the roof is made out of endoderm. We would call this midline endoderm. And what is noticeable here at this early stage is that the endoderm makes these outpocketings that are called pouches. And these pouches form in an anterior to posterior direction, and they contact the ectoderm of the skin at a sort of depression that we call a cleft. So this is a pouch cleft contact, and you have them in each of the pouches. They are not sufficiently developed here, but in the end you get such a pouch cleft contact between each of the pharyngeal arches. And to show you how this looks like in a section, so if you make a section in this area, so we have a cross section so perpendicular to this plane, we see here lined by green, apologies, the endoderm, and at both sides these outpocketings or pouches that contact the ectoderm at the cleft. I also show you a picture of the skin because the ectoderm, as you will understand, plays an important role in the story. The ectoderm at this point is composed of two layers, a basal ectodermal layer and a superficial layer, which is also called the periderm. Now, periderm is a separate story. I will not go into details where that comes from and its fate and so on. But just for the time being, the periderm is the outer layer of the skin, the outer ectodermal layer, although that can be discussed. Okay. So what is important is that these pharyngeal teeth, they are located, as I say, deep in the pharynx. They are attached to the last pharyngeal arch, right? And just in front of them, you have the last pouch, pouch 6. So this is pouch. This is here. You can see here the pouch 6 cleft pouch contact. There's a lumen forming already. But if you go closer, and it's not the same animal, but you can see in better detail the endoderm here in the middle, and then on each side these pouches that extend to the ectoderm, and it is these pouches, in fact, that will open into gill slits. So they will sort of split, a lumen will form, and they will form the gill slits. But what I wanted to point out here is that the two germs, they develop at just behind these pouches. So they develop from this endodermal epithelium here, but just behind the last pouch. If we go a little bit later, this is a 3-day-old zebrafish, 3 days post-fertilization, and here the teeth have already started to develop. So each of these little clusters of cells is one individual tooth germ, corresponding to the schematic that I showed you before. Okay, so this is a lot of introductory stuff to come at the point where I wanted to come. Zebrafish have the advantage that you have a lot of tools to work with. So we used we used transgenic lines, we used different mutants, and we did lineage tracing experiments. And I will summarize what these experiments and what these data gave us. First of all, they showed that pharyngeal teeth indeed develop from endoderm. We have here a transgenic line where GFP, so green fluorescent protein, is expressed under the promoter of the SOC17 gene, which is an early endodermal gene. And you can clearly see here, this is just behind pouch 6, so we have the endoderm, and we have the two germs developing clearly from SOC17 positive endoderm. This is just the corresponding histology to show you where we are. So that's one thing. All right, the pharyngeal teeth develop from endoderm, yes. However, the exciting thing, then we got really excited, was when we used another transgenic line, one that labels or is specific for the periderm, so for the outer ectodermal layer. And this is again a cross-section through an embryonic zebrafish. You can see here the outer layer of the skin, clearly in GFP. This is a keratin 4 transgenic line. Keratin 4 is an intermediate filament specific for periderm. And we were absolutely excited when we saw this. I hope you can see here this thin line which delimits the endoderm, and the endoderm where the teeth develop, that's where the arrows point to, it's clearly covered by something that is very similar to periderm. So we were very enthusiastic with it. Okay, this is clear evidence that somehow the periderm must come in. So we started to search where the periderm comes from. And what you see here are sections or parts of cross sections at the level of pouch 2, so much more anterior. And we saw that the periderm starts to invade pouch 2. We saw it with our transgenic line. We saw it also with lineage tracing. So you can clearly see here the outline of the pouch, the periderm that is invading. And so, again, we got very enthusiastic of that, but then we realized that the periderm is this invasion or this expansion into the pouches is arrested. It's not going to the midline where the teeth form. In fact, the periderm invasion is arrested here. But what we saw was another population of cells in the midline, so clearly also labeled with this keratin 4, periderm-like, and there are other markers that we used. They look very similar, but this population is developing endogenously. It's not coming from outside, it is endogenously formed. So, yes, periderm expands into the different pouches, but it's periderm-like cells that do not come from outside that cover the prospective tooth-forming epithelium. And that was, of course, a little bit disappointing. We had hoped for another result, but that's how it is. And another thing that we observed is that despite the fact that the periderm does not penetrate deep enough into the pouch to reach the tooth-forming area, what we could establish is that the pouch-cleft contact needs to be formed, this pouch 6, just anterior to the teeth, it needs to be formed before you can have any teeth. And we quantified this also. We have lots of quantitative data that supports this observation. So to take all these data together, we made this interpretative scheme. It's a kind of a slice now. It's not a thin section, but it's a slice through a zebrafish embryo. Now, in this case, the endoderm is in yellow. The tooth germs are developing from the endoderm. From the endodermal epithelium. Of course, I should add, they always form in conjunction with mesenchyme. You cannot have a tooth without the underlying mesenchyme, of course. But the endoderm is covered with periderm-like cells. And the third observation with it is that, yes, periderm invades the pouches, but only in the distal parts, and it connects with this periderm-like layer on top of the teeth. Oops. Okay, so all right, periderm only has a limited expansion. It's not covering the teeth. You need some periderm-like tissue. So the idea was, or the hypothesis was, maybe this is something specific for teleosts. Teleosts are advanced ray fin fish, advanced actinopterygians. They have a very specific early development, of course. They have very yolky eggs. They have partial cleavage, they have a special pattern of gastrulation, of endoderm formation, and so on. So we were wondering what would happen if we looked at more, or at representatives of lineages that branched more basally from this clade. And so the question was, is this periderm behavior specific to teleosts, or do we see something of a much more vast migration of periderm in non-teleost actinopterygians, for example in sturgeon. And that's what brought me here to Prague, this question, right? So yes, we engaged in a study on sturgeon. Let me first show you the skin, the ectoderm in sturgeon. We are, of course, limited in terms of what we can have as tools. We don't have transgenic lines, but we can do lineage tracing, same sort of lineage tracing that we use for zebrafish and which is commonly used with big success here in Robert's group. Yes, okay. So at least we have an at least bilayered ectoderm. That's fine. The superficial layer. Well, you can discuss if it's a periderm or not a periderm. There is a discussion in the literature. Just for simplicity, I call it periderm. And then we, of course, looked what's going on in the dentition. Now, some of you correctly will say, yeah, but sturgeons don't have teeth. That's true. The adults don't have teeth, but they develop early in development. They develop quite a number of sets of teeth that are represented here in this cartoon. They have 6 sets of teeth. And first we looked at the oral teeth, so on the jaw margins, and we were very excited to see that the 2 germs that develop, whether it's on the upper jaw or on the lower jaw, were indeed nicely covered by invading periderm. So that looked very promising. Then we looked at the gill slits, and here we see successive cross-sections through sturgeon embryos at the level of the different pouches. The cartoons are next to the pictures. So what you see here is, this is of course the yolk here, this is the pharynx, and then here is pouch 2, which is almost open to the exterior here, or is open at another sectional level. But the pattern that we saw in pouch 2 was the same in pouch 3, in pouch 4, forming pouch 4 here, and later pouch 4 here. What we saw is that we observed invasion of periderm inside the pouch, yes, but again limited. Again limited to the distal side, the distal part of the pouch, and certainly not up to the midline, where, for example, also pharyngeal teeth form. This is just a blown-up figure of this periderm within pouch 3. In this case, it is, as I said, only in the distal parts of the pouch, and it is also discontinuous. So what about pharyngeal teeth? And well, I think I've given the answer already. For the oral teeth, we did see cover by periderm, but we didn't see it for the pharyngeal teeth. For example, here the palatopterygoid tooth patch. Here you can see that the periderm is arrested here, and the tooth develops here. Same for the hypobranchial teeth. There's nothing to see here. You can see it at the outside in the skin, but you cannot see it in the place where the hypobranchial teeth form. Now, does that completely reject our hypothesis of an ectodermal influence inside the mouth? No, we don't think so, because first of all, over the studies that we did, we noticed that vertebrate teeth always develop from a stratified epithelium. You will never see a tooth forming from a single layered epithelium. So the epithelia are always stratified, and if it's not periderm, there we see a lot of apical differentiations, let's put it this way, in different representatives of vertebrates. So the surface layer appears to have an ectodermal signature, whether it's as a periderm, periderm-like, or as what we, not us alone, but the term has been coined before, ectodermalized endoderm. So that leads us to my first set of conclusions. I think we can say that the homology between dermal denticles and teeth is well established based on quite a number of arguments. That epithelium with odontogenic competence may have invaded the oral pharyngeal cavity to generate teeth in gnatostome vertebrates, or alternatively may have transferred the competence to internal endodermal epithelium, and I think there's more evidence now for the second possibility. That pouch cleft contacts play a crucial role in pharyngeal tooth initiation and for periderm invasion. And a cover of periderm-like cells required for pharyngeal tooth formation in zebrafish is just one way of how stratification of tooth-forming epithelia is achieved. In the beginning of the talk, I alluded to the fact that early in gnatostome evolution, or if you look in the basal part of a lineage, the distribution of teeth is always much more vast than in the more derived species of a lineage. So we sort of evolve from many to few. The examples that I want to show you come from different lineages. Sharks is a bit of a difficult sample to discuss, because shark teeth are not attached really to ossified structures, obviously. So we find them in loose amounts, and it's difficult to assign them to a particular anatomical structure. So just moving to the actinopterygians, if you look, for example, to these two teleosts, this is an elopomorph, so a very rather basal teleost. This is a very advanced teleost, and in the more basal teleost, teeth are really distributed all over the different pharyngeal arches. The mandibular arch is not even shown here, but the hyoid and all the branchial arches carry teeth. This is much less the case if you advance in the lineage to more derived species. Usually you have sets of teeth retained on the jaw margins, so the upper and lower jaws, and on the pharyngeal, usually the last pharyngeal arch. So you have this reduction in distribution. You also see this reduction, and maybe this is familiar to you, as you go from reptilian ancestors to mammals. We have only one series of teeth on our jaw margins, but that was different in reptilian ancestors. Now, this is not a direct reptilian ancestor, but it's just to show you how widespread the teeth are here in the roof of the mouth cavity. So again, very clearly a reduction, loss of teeth on different pharyngeal arches. And the question is, of course, how was that achieved? This is work that was not done in our group. It's work from Jan Gibert and his colleagues. And they observed that, they worked with retinoic acid, and they observed that if you inhibit retinoic acid formation, retinoic acid is a metabolite of vitamin A, and in the last step, the transition to retinoic acid is catalyzed by an enzyme called retinaldehyde. And what they did was blocking this retinaldehyde. So they blocked the formation of retinoic acid, and in zebrafish. And you can see here the different branchial arches. This is a control here. You may notice in this blown up inset the presence of the pharyngeal tooth, or even teeth, at least one here. But inhibiting endogenous retinoic acid, they lost the teeth. However, even more interesting is when they added retinoic acid, so when they did the opposite, when they added retinoic acid, they could expand the dentition that is normally only on the last pharyngeal arch, as I told you. Like here, these are the more anterior arches. This is a control situation. And when they added retinoic acid, they could expand the dentition forward. Is what you see here also in these pictures where they use the transgenic line. So adding exogenous retinoic acid is sufficient to induce anterior ectopic tooth development in zebrafish. And then it's a logical step to think that changes in the location of retinoic acid synthesis correlate with evolutionary changes in vertebrate dentitions. So tinkering with the retinoic acid may have changed the distribution of teeth in the oropharynx. However, there was a sequel to this. There is a sequel to this story. They did the same in Medaka, and they said no, inhibition of endogenous retinoic acid formation does not prevent pharyngeal tooth initiation in Medaka. It's very different from zebrafish, from which they concluded that pharyngeal tooth induction is not dependent on retinoic acid in Medaka versus in zebrafish, which was kind of suspicious given the very conserved nature of tooth development in vertebrates. So what they didn't take into account, in our view, is pouch cleft contacts. In 2006, a paper was published that showed that if you inhibit endogenous retinoic acid, you actually prevent pouch formation. So the idea was maybe the teeth for the formation are not directly dependent on retinoic acid, but via the pouch. If you don't have a correct pouch formation, you don't have a correct pouch cleft contact, you block tooth formation. And even more suspicious is that, or perhaps supporting this idea, is that when they did the experiments with Medaka, they did the inhibition after the last pouch had made contact with the ectoderm. So the pouches were completely formed. So we are now pretty much also engaged in testing these ideas, and it seems that Medaka is not different from zebrafish if you respect the correct timing of the pouch cleft contacts. Besides of that, we also redid the experiments with adding retinoic acid, and it's very remarkable also that we observed supernumerary teeth exactly here at contact with the ectoderm. Also something that we will further take into account. So, conclusion from this part: pouch cleft contacts, or there is quite some support again for pouch cleft contacts playing a crucial role in pharyngeal tooth initiation, and the distribution of teeth within the oropharynx that was once vast in early representatives of lineages appears to be controlled by retinoic acid signaling. Now I want to go briefly to the last part of my talk. Probably many of you know that if you look at a non-mammalian that has teeth are replaced throughout life. We don't have this potential. When we lose our permanent teeth, unfortunately, it's done. We cannot produce a third generation of teeth, unfortunately, but all non-mammalians can do that. At least the ones that have teeth. Not birds, of course, they don't have teeth, but other non-mammalians can do that. And you probably are familiar with this aspect from sharks that have these batteries of teeth that sort of roll out of the mouth to produce successive generations of teeth. But it's also the case in not just in chondrichthyans, but also in osteichthyans. Again, pictures from zebrafish. And this little epithelial strand here is in fact the start of the formation of a replacement tooth, which is then in a more advanced stage here. So we have been intrigued by the question, of course, what is the mechanism behind? Why do non-mammalians, why are they capable of continuously replacing their dentition, and why has it been lost in mammals? So the hypothesis that we have proposed now, oh my God, almost 20 years ago, was that there could be a niche of a set of adult epithelial stem cells that somehow reside in this epithelium and that generate the next tooth. The hypothesis was proposed based on a number of other cycling organs in sarcopterygians at least. For example, in intestinal crypts, the epithelial stem cells are located deep in intestinal crypts and are responsible, of course, for the renewal of the cells of the villi. Another example is the hair follicle, where the bulge region contains stem cells that are responsible for the cycling of the hairs. And then, most interestingly, stem cells were also described in rodent incisors. Now, rodent incisors are a peculiar type of teeth. They are not replaced, but they are continuously growing. They are worn off at the tip, but they are continuously growing at the base. So you need these stem cells to produce at least the epithelial derivatives necessary for the tooth. And there's also mesenchymal stem cells, but I will leave that aside. Okay, so we set out to test this hypothesis. Now, stem cells, what are the characteristics of stem cells? For a long time, it was thought they are slow cycling, so they proliferate. They don't proliferate much. There's a long time between two replication events. But more specifically, they give rise to. They show self-renewal, which means one stem cell divides. One daughter cell will make the new stem cell. The other daughter cell will start to proliferate and differentiate into a particular lineage. So we use this slow, or not just us, but many people have used the slow cycling character of epithelial stem cells to identify them in tissues, and we have done the same. This is work from a previous PhD student from us. And he used different species. He used apologies. He used a shark species, a cat shark, but he also used some actinopterygians, a polypterus, a bichir, so a basal actinopterygian and a teleost. And he looked for slow cycling cells. So what you do is you label the animal. You inject the animal with BRDU, which is incorporated in the DNA, and if the cell doesn't proliferate, it retains the signal for a very long time, for a chase time, as we call it, for months, weeks, or months. So he did this for a shark. This is a section through. There's a cartoon here. This is this long epithelial strand that produces sequentially the teeth that will eventually come out here. This is called dental lamina. Sharks have a permanent dental lamina, so this structure persists throughout life and forms one tooth after the other. In actinopterygians, we have a different situation. For example, in Polypterus, the replacement tooth develops really very close to the functional tooth from the superficial epithelial layer of the functional tooth. And something similar happens in some. So what the PhD student found, he found indeed label-retaining cells, putative potential stem cells, deep inside the dental lamina of the shark with the permanent dental lamina, indicated here, or the position indicated here by an asterisk, but he did not find them in actinopterygians. And in a way, it's understandable because teleosts, or also, both in the case of the bichir and in the bichir, we simply don't have a dental lamina. As I said, the tooth is really completely adjacent to the functional tooth. But even in other teleosts, there is at most a transient dental lamina that we call a successional lamina. So something that is transitory, that is transient and disappears after every cycle. The other thing that could explain why we don't see these label-retaining cells here, and therefore no evidence for stem cells, is that the stem cells would have to make a very unlikely migration, because the next stem cells would have to move from here to here for the next and the next tooth generation. So together, this suggests that yes, maybe epithelial stem cells are present in chondrichthyans, but not in actinopterygians. Of course, that was criticized, so we continued a little bit along these lines, and we shifted to Medaka for another advanced teleost for this, and we looked at telomerase. The idea behind is that if you have cells that have to support lifelong renewal, you can imagine that they want to protect their DNA from breakdown at each replacement cycle. So you would expect they express telomerase, which is the enzyme that catalyzes additional telomere repeats. So we looked at the expression of telomerase. This is the catalytic component of telomerase. Here in immunohistochemistry, so you have to look at the brown dots. And what we observed is that in the epithelial part, certainly at the early stages of tooth development, we could not observe any telomerase. In fact, the pattern of TERT was very complementary to the PCNA, which is a marker for proliferation, but that's another story. We also looked at typical stem cell markers like LGR5, which is a bona fide stem cell marker. We looked at OCT4, which is a pluripotency marker. And without going into details, we did observe, we had signals, but not in the developing tooth germs. Yes, we had ample signal in the overlying epithelium, but not in the developing tooth germs. So we concluded from the previous and from this study that neither a dental lamina nor epithelial stem cells appear to be required for tooth replacement in teleost. Bit upsetting, maybe. So, what could be? What could play a role? Well, we shifted our attention to two structures that are rarely considered in this context, and that is the vasculature and the innervation. Why did we look at the vasculature? Because the vasculature is so prominently present in the dentitions that we looked at. For example, you look here. On top of a zebrafish dentition, these are the tips of the teeth here in gray, and the red in between is a huge sinusoidal blood vessel. And you can see it also in a cross-section. These are cross-sections through the teeth, and here are these huge blood vessels full of blood. That was one observation. Another observation was when we looked at the replacement, we observed that from a larger blood vessel, it looked as if there was a small branch branching off, going to the place of replacement. So we thought, okay, let's try to block the formation of these small vessels and see what happens. That's another PhD student who did this work. We used, we blocked VGF signaling, vascular endothelial growth factor signaling, which is required for angiogenesis. So it blocks the formation of blood vessels, and what we observed was the following. We observed not that tooth replacement was stopped, tooth replacement went on, but it clearly delayed replacement tooth development, and that can be seen in this graph, which I will briefly guide you through. Each of these bars represents a category of functional teeth. So these are very young functional teeth, and we go here to the very old functional teeth that are close to be shed. And in each bar, the different colors show different stages of replacement teeth. And you don't have to know the details, just look at the pattern. As you go to the more mature, the older teeth, you can see that the replacement tooth is now in the oldest category, is of a very advanced stage, the dark green, not the light green, not the blue. But if you look at a similar situation in the experimental animal, you can see that even in quite old teeth, there are still very young replacement teeth, leading, as I said, to this conclusion that tooth replacement does not require de novo formation of blood vessels, but that blocking VEGF signaling delays replacement tooth development. That's about the vasculature. About the innervation, I have to leave you completely in the dark because, and it's also a bit dark, it's black and white, yeah. It stems from a paper from 1994, and this is, to my knowledge, the only paper that had really very important data on innervation and tooth replacement. What these people from Sweden did, they used cichlid fish, and they cut the ramus alveolaris, so a branch of the trigeminal nerve that serves the lower jaw. And they looked after 100 days. So this on the left is the control, nice replacement teeth sitting ready, and nothing on the innervated side. So clearly the innervation, plays an important role. But other than that, and we have never been able to start studies on the innervation, but it's certainly something that is worth exploring further. So this brings me to my end conclusion. We had already the other conclusions. The lifelong replacement of teeth may well differ between chondrichthian and osteichthian lineages, consistent actually with recent ideas about the specialized character of extant chondrichthians. And with that, I like to thank all the collaborators in my, let's say, old lab. First and foremost, Eckhard Witten, and also two wonderful postdocs, Veronika Oralova and Joanna Rosa, who did all the zebrafish work on the periderm, Daria Larionova, who is an excellent electron microscopist, our technician, but also other people. I had not enough space for all the photos. Of course, I'm very thankful and grateful to my new lab here in Prague, under the guidance of Robert, and with two wonderful PhD students, Agata and Tomáš, and very nice discussions also with Vladimír Soukup. I acknowledge funding both in my home university and from the Gatcher, international collaborators, people that gave us the transgenic lines, and here in Prague that give us the sturgeon embryos. And I want to thank you very much for your attention.